Hello everyone, my name is Alan, and today I'm going to present effects of virgin micro and nanoplastics on fish, trends, meta-analysis, and perspective. And first, let's talk about what is plastic. As we know that plastic is a durable, inexpensive, and lightweight, and have isolation and resistance property that offer many medical, societal, and technological benefits. And for those reasons, the global production of plastics has increased from 2 million tons in 1950 to almost 350 million tons in 2017. And production is expected to reach 1,800 million tons in 2015. And in parallel, 60% of all the plastic produced from 1950 to 2015 were discarded in landfill and in the natural environment. And the worst is that the 3.4 billion tons are expected to be disposed of in the next 30 years. And with an estimated 2 million tons of plastic waste flowing from river to oceans every year, the increasing occurrence of plastics in the aquatic environment along with its increasing production makes plastic pollution a major ecological and societal concern today, as I've just mentioned. And this concern is even higher when considering smaller plastic. For example, the microplastic, which is defined typically as particle below 5 millimeter, or defined as particle below 100 nanometer, as they can translocate in tissue and propagate up the food chain. And what is nanoplastic? As I've just talked, the microplastic is defined typically as particle below 5 millimeter, and the nanoplastic is defined as particle below 100 nanometer. And we can see this figure. 1 millimeter equals to 1,000 micrometer, and while 1 micrometer equals to 1,000 nanometer. And in this figure, we can see here has the parameter which has size and concentration, and the exposure has water, food, and type has 4. It has the water mass, the water particle, and the food mass and the food particle. And this figure summarizes all the microplastic and nanoplastic size and concentration class used in this review. And the MISC means the miscellaneous class, which means when a polymer size included 2 or more size, or when polymer concentration was not possible to standardize. And here are the occurrences we are facing now. As I just said, the environmental plastic pollution is a major ecological and societal concern. And over the past decades, a broad range of laboratory and experimental studies have complemented field observation in the hope of achieving a better understanding of the effects and impacts of microplastic on the diverse organism. And however, the plastic pollution remains challenging to monitor in the environments and to control under laboratory conditions. And plastic particles are often naturally or experimentally contaminated with diverse chemical pollutants. Therefore, our understanding of the effects of virgin NP is in freshwater and marine fish is still limited. And later I will perform a systematic review of the latest literature on the effects of virgin plastic on fish under laboratory conditions. And experimental studies reveals that the toxicity of virgin plastic on fish should be more systematically evaluated. And using a laboratory-based method and aims toward a better understanding of mechanism of the toxicity to fish. And by the way, the plastic was artificially added to the fish in order to examine the effects on the plastic to the fish, which was not naturally ingested by the fish. It is added by the scientists in order to study the effects to the fish. And so far so good, as I hope I will convince you that we should recycle as much as we can go green. And this is the literature research. It's the paper that published before 2019, and the studies focus on the effects of plastic on fish and using all this database. And the paper doesn't generate the scientific data. It summarizes the data generated by the other people. This study only chooses the study that is based on laboratory experiment conditions. And the experiment is the study correspond to the experiment performed under the laboratory conditions. And the study examined at least one toxicological effects of direct exposure to virgin plastic on fish on the larval, juvenile, or adult stage. And the study also has included the no plastic control treatment, which has the original group and control group. And the results of the study are supported by appropriate statistics. And we can see that this is the general trends in the experimental research. And this is the X layer is a year and the Y layer is the number of studies. And we can see that the studies has increasing. And this is the general trends in the experimental research of the effects on plastic on fish. And because this paper is the summarize all the experimental condition, the percentage here is a pie chart above means the statistic of the experiment, not the percentage of the fish effects by the plastic. And we can see the B2G on the pie chart respective proportion of fish, water environment, feeding diet, life stage, type, shape, and exposure wave to fish. And we can see the statistics of the experiments in the freshwater have the most, and in the amphibians have the less. And this is which means this is snails. And with all the charge gathering, we can get the exposure pathway to fish. In the water have the most, and along food has the less. And this is the ecotoxical effects of virgin plastic on fish. And the white part of the pie chart is the total number of the fish, and the color chart, and the color part is the number of fish which has affected. And the red one here, the red one means the fish are sensitive to plastic, while the green one is less sensitive. And here I enlarge two of the pie chart. As we can see, the total diversity and the probacteria has a most affected, and the antibacteria has less effective. And the immune system, almost all the cells of immune system, chemokines and cytokines and other molecular actors are almost not effective. And my conclusion is, even in the worst case scenarios of plastic pollution, plastic will likely remain a minor fraction of the other microparticle naturally present in water.